And we're on. Perfect. Thank you, Mr. Hoeschen, for joining me today. We are going to be talking about the project IMMPRINT. You are the project coordinator, so if we could start with the first question, if you could introduce us your project, please.
Yeah, thank you very much for giving us this opportunity, especially me as a person, but it's not me, it's a team that is coordinating that, and also that's performing the project. IMMPRINT stands for Integrated Molecular Imaging for Personalized Biomarker-Based Breast Cancer Characterization and Treatment. And it's one of the projects that were elected during the first call of Pianoforte. And it is under the topic 2, which is individualized diagnostic and therapeutic procedures with regard to optimization of the benefit-risk ratio. And the idea of IMMPRINT in principle is that we develop new imaging modalities, new imaging methods that can be used as like kind of biomarkers, in this case for breast cancer patients, to characterize the breast cancer and optimize the treatment based on the sensitivity of the breast cancer to ionizing radiation, as well as of the healthy tissue of the patient. We want to individualize the treatment of the patients in the future with our approach to spare healthy tissue from ionizing radiation and to improve the effect, the beneficial effect of radiation treatment to those patients by individualizing what we treat, how we treat, what kind of radiation we use, and things like that. To do so, we develop imaging methods. And these are, on the one hand side, dark field, and on the other hand, X-ray fluorescence-based imaging procedures. And we do that in vivo, so we really characterize at the end of the project, hopefully really in vivo models, so in mice or something like that, the tissues. We certainly start with in vitro measurements and develop the systems for in vitro measurements first, because we need to be sure that everything works out before we go into in vivo studies.
Yes, see, thank you. Can you describe your consortium and explain how you selected and engaged your partners?
Yes, for sure. Yeah, we have a really great consortium to my point of view. So as you have heard, Otto von Guericke University in Magdeburg is coordinating the project. By the way, I should say we have a very close collaboration with another project within this first call in topic 2, which is the Imagomics project, which does a lot more biology than we do. But our methodologies will be implemented in there, and vice versa. Their biological findings will be implemented in our project. So that's a way on how we decided on our second partner, that is the National Public Health Center from Hungary in Budapest, the groups of Geza Zafrani and Katalin Luminski. And then we have the we need, for our imaging methods, we need a group that can do synthesis of nanoparticles, because we need nanoparticles. So that is done by the Heinrich Heine University in Düsseldorf, where the group of Christoph Hoeschen is working on that. And for our imaging tasks, a lot of things we can do here in our institution, but the development of specific detectors, that's something that needs additional competences. And I had worked in previous EC-funded projects, which I had the honor to coordinate together with two groups, Joseph Stefan Institute in Ljubljana in Slovenia and the CSIC Institute in Valencia in Spain. And these are the other groups that are involved. And then because we, as a coordinator, in this case, are only an affiliated entity, our main principal partner in Pianoforte is also involved, that's Euramed. They have to do the finances and support us with administrative stuff. Yeah.
Oh, thank you. What key results do you expect from the project? And how do you foresee these outcomes impacting the EU community?
Well, the main idea, as I said, is to develop imaging tools as biomarkers, right? And with these biomarkers, if we can evaluate them in some years, obviously not during the time frame of our project, but in some years, really in patients in vivo, then we could optimize the treatment for the patients of breast cancer using ionizing radiation for radiation therapy treatments and personalize that much more than what we can do today. With that, we want to do two things, basically. First, we want to control the breast cancer better. And secondly, we want to avoid detrimental effects on the breast cancer patients. And to do so, we will develop these imaging methods. We will also look for certain biomarkers that are already there and look how they are interlinked with each other. And if we have developed these tools, I mean, then they can be produced by European medical technology companies and nanoparticle companies and marker companies, so chemical companies and so on. But the main thing, obviously, is to improve what we can do for the patients in these fields. So especially women that suffer from breast cancer, yes, in our case. So the idea about that is that we personalize the treatment, that we personalize breast cancer medicine, and by that improve cure while reducing detrimental side effects. And I think that's very important. And obviously, we are working together closely with other projects. I mentioned already Imagomics, but there are others where there's also some interlinks in optimizing radiation therapy approaches. And what we do can fit into projects that develop, let's say, more technological radiation therapy approaches. So our part is on the imaging as first steps, and then this can be used. So the long-term perspective is really to have a European-wide integration of our method and tools into the clinical routine, to implement clinical strategies for that, and to commercialize, obviously, also the technologies that involve diagnostic and therapeutic methodologies.
Thank you. Last question. As your project has been running for about a year now, what would you highlight as the most significant achievements so far?
Okay, we already have achieved a number of things. I'd say we are more or less working along the lines of our proposal output. So one thing that is obviously very important is that we know how to build the imaging system, right? Because we want to integrate dark field and X-ray fluorescence imaging. So we need to know how that looks like. So we have, meanwhile, simulations, we have cut drawings, how the systems would look like. We have established a new source for X-rays here in our lab. Then the next important part on that is if this system should be running, we also need the detectors. And also the detector configuration, we went in with a certain idea. This has now been optimized and already been designed. So they start already with the configuration of the system now they will buy some certain components and then optimize them for our projects and stuff like that. So that's already also ongoing, and it's in time. The next important thing is, as I mentioned, for the X-ray fluorescence, we need nanoparticles. And so we need to synthesize nanoparticles, which we can then ligand to biological probes. And actually, we also succeeded already, not we as OVGU, but the project, so the Heinrich Heine University in that case, succeeded already in synthesizing first nanoparticles. There's a design factor for how large they will be, how they will look like. And also there is a first general approach on how to link them to potential biomarkers. these biomarkers are on, that is the fourth part that we already also worked on very successfully, that we have certain biomarkers identified which can be linked. This step will then follow, and step by step we will come up with the imaging part. So we are kind of in time, and all these works together, otherwise it would not work. So therefore it's difficult to highlight just one most important one step, but I think all partners has provided what they had been promising to provide until now. So that's basically very nice.
That's great news, I would say.
Yes, very nice for a coordinator.
Yeah. Oh, thank you very much. Thank you for making your time and enlightening us about your project. I really appreciate. Are there any last words that you would like to add?
Yeah, I think, first of all, we should say thank you to Pianoforte to make this happening. Then I also would like to mention Euramet, who did a lot of things together with all the other platforms in designing the topics for the different calls. This is for sure coordinated within Pianoforte, within the work package too, but the platforms really did a great job in working on that. And so this interesting topic, helping hopefully a lot of patients, is coming from that, and it's bringing together, and I think that's most important, the science of many, many different disciplines. So it's a very interdisciplinary project, and we really enjoy that a lot. And that's also true for the cooperation with the other projects running in Pianoforte. So I think I should mention that. Thank you.
Oh, thank you very much.